Matteo Bonazzi, qui travaille sur Coxiella donc à Montpellier, donc c'est juste à côté, et il développe en fait des mutants de Coxiella donc la forme virulente, pour essayer de comprendre la forme virulente, et il va nous présenter aujourd'hui ses travaux. Voilà, je vous remercie.
Thank you very much for inviting me here today to show what we've been doing so far with Coxiella. And so I'm Matteo Bonazzi, and I arrived in Montpellier last year to set up and coordinate a team of cell biology and bacterial infections at the Centre d'études d'agents pathogènes et biotechnologie pour la santé. It's a pretty complicated name, but you can just call it CPBS, which is a mixed unit of CNRS that, as the name suggests, is involved in the study of bacterial infections. And since last year, we're in a very nice and new building in a CNRS campus in Montpellier. So what we study in the lab, what we're interested in the lab, is the study of the host pathogen interactions that regulate the infection by intracellular bacterial pathogens. And the reason why we're interested in this is because we find it interesting that at some point during the coevolution of bacterial pathogens with their host, a conspicuous actually number of bacterial pathogens decided to, well, actually I don't know if they decided or not, but they evolved ways of internalize the host cell. And there are a couple of ways basically the bacterial pathogen can do so. One is called the zipper mechanism, by which the bacterium has evolved proteins that interact with receptors at the surface of the host cell. And the interaction between the receptor and the ligand at the surface of the bacterium mediates the internalization of bacteria. Other bacteria chose a more strong way to enter cells, and is by injecting bacterial effectors in the cytoplasm of the host cell. This induces the rearrangement of the actin cytoskeleton and the engulfment of bacteria inside the cells. Now, entering the host cell presents several advantages in terms of bacterial infection, because first of all, bacteria are protected from the extracellular antimicrobial activity of the host. Inside the cell, bacteria can access nutrients away from the competition of other bacteria that are present outside the cell. And last but not least, actually entering some kinds of host cells could actually be a pretty efficient way of getting transported inside the infected organism and access organs that are far away from the initial site of infection. The problem is that whatever the host cell internalizes gets delivered to the endocytic pathway, and it's fused with the lysosomal compartment, where normally the cell degrades everything that is taken up, because this is the way the cell eats and gets nutrients. The problem is that bacteria do not want to fuse with this endosomal compartment, because they want to survive and actually replicate within the cell. So what is interesting is that a wide number of bacteria has developed different ways of interfering with a wide number of host signaling pathways to perturb the endocytic maturation pathway and generate specific niches for their intracellular replication. Now, this is a very, very simple scheme that only illustrates a few of these examples. We have a number of bacteria that chose the most straightforward way, which is to lyse the vacuole they have been entrapped in by means of bacterial toxins. And now bacteria are freely swimming in the host cytoplasm. And they can interact with the actin cytoskeleton of the host again to form these so-called actin comet tails, move around in the infected cell, push the plasma membrane looking for the next neighboring cells to infect and spread the infection. This is the case of, for instance, Listeria, Shigella, and Rickettsia. Some other bacteria have developed a more subtle way of interfering with the host signaling pathway by remaining actually within the internalization vacuole, but secreting bacterial effectors in the host cytoplasm to subvert a number of mechanisms and generate hybrid compartments that are suitable for bacterial replication. Now again, to give a few examples, Brucella fuses with membranes of the endoplasmic reticulum to deviate from the normal endocytic maturation pathway to generate these tight-fitting compartments that are compatible with bacterial replication. Legionella merges again with the endocytic reticulum, but in this case to endoplasmic reticulum, sorry. In this case to generate a very big vacuole where bacteria can replicate. This is also similar to the case of chlamydia that fuses with Golgi membranes. The different strategies involved by intracellular bacteria are really diverse and very specific. And then there are other bacteria, such as Salmonella and Shigella, for instance, that stop endosomal maturation at different steps. And then there's this last case of Coxiella that actually is pretty unique because it doesn't really try to interfere, at least not evidently, to the normal endocytic pathway, and actually resides in a very large vacuole that is biochemically very similar to a lysosome, and actually the low pH is favorable to bacterial replication, which is very resistant to lysosomal degrading enzyme. So this gives a bit of a quick overlook of what intracellular pathogens do. Now, it's important to understand all these mechanisms of subversion of the whole cell if we want to develop targeted strategies to interfere specifically with the infection by a determined bacteria. And on the other side, actually, the fact that bacteria have evolved all these strategies could be useful if we want to use bacteria now as probes to challenge the whole cell and uncover novel mechanism of the whole cell outside infection. Now, to study the host-pathogen interaction, we are working in the field of cellular microbiology, which is a relatively new field, about 10, 12 years old. And cellular microbiology actually uses our knowledge of cell biology to understand bacterial infection. And as I just mentioned, you can use microbes to investigate cell biology. And all this is made possible by the use of molecular biology to generate all the tools we need for our research. Now, when cellular microbiology emerged, it was immediately obvious that this was a remarkable advantage, because instead of studying and investigating bacteria as an isolated unit, we can now study bacterial infection in the context of the infected cells, and we can also study and target, actually develop host-targeted therapies to fight infections. Now, what we are trying to do in Montpellier, we're putting a lot of effort in trying to merge high-content screening with cellular microbiology, because we think these are really powerful techniques, but they're really time-consuming. So what we're trying to do is to develop a number of screens to get the most out of every single experiment that we do. And the idea is to get one day to what we can call high-content cellular microbiology. And to do this, we chose to work on Coxiella burnetii for a number of reasons that I hope will be obvious in a few minutes. So Coxiella, as some of you might know here, because you're always working on Coxiella burnetii, is a gram-negative obligate intracellular bacterium. It infects farm animals, either directly or via via ticks. And the bacteria accumulate in animals, and especially in the placenta of infected animals. And during delivery, bacteria get spread in the environment. Coxiella is extremely resistant to the environment, so it can contaminate hay and dust in farms. And people working in these environments, in farms, are extremely exposed to Coxiella infections. In humans, Coxiella gives rise to an acute disease that is associated with pneumonia and hepatitis, that doesn't have a very high mortality rate, but it sometimes can convert to a chronic disease that is more serious, let's say, and has a very high mortality rate and gives rise to endocarditis and other symptoms. As I said, Coxiella is the causative agent of Q fever. Q fever is considered an emerging zoonosis. And these are numbers of reported cases of Q fever in Europe and France, and as you can see, the numbers tend to increase with years. This is also, to be honest, associated with the fact that within the years there's more awareness of Q fever, and the ways to detect Q fever have improved. So this is also reflective of these advances. And these are a few numbers of cases of Q fever in France from 85 to 2009. I think the most notorious case of a Q fever outbreak is the one that occurred, actually started in 2009, 2010 in the Netherlands, and is actually still ongoing. And to give you an idea of the severity of this disease, before the outbreak in the Netherlands, we had like about 180 cases reported per year, and during the outbreak, these cases raised to 2,361, indicating that this is, during an outbreak, this is a very severe infection. So as I said, Coxiella is also extremely infectious. due to some military tests back in days where these were possible. It has been determined that in human the infectious dose of Coxiella is as little as 1 organism, and so this Coxiella is really extremely infectious. But for us that wants to work with Coxiella in a lab, the advantage that this bacterium is present in 2 phases. It's called phase 1. That is isolated from infected animals and patients. And upon several passages in vitro in a laboratory, there's a spontaneous conversion of phase 1 to phase 2. Now this conversion has been associated to a big deletion of approximately 26,000 base pairs in genes that code for the bacterial LPS, which is then severely shortened in the phase 2. And this has been associated with a severe attenuation of virulence of phase 2 in animals. So if we inject phase 2 in animals, these will not give rise to an infection. However, inside culture cells in the lab, these 2, phase 1 and phase 2, replicate with similar kinetics, and the different steps of the infectious cycle are very similar. So we can actually pretty much use phase 2 as a model to study bacterial infection of phase 1 without the risks associated with phase 1, and especially with the advantage that if we want to work on phase 1, we have to be in a P3 environment, whereas phase 2 is manipulatable in a P2 environment, which really facilitates experiments. Coxiella also has a unique intracellular adaptation, as I mentioned before, because it's the only known bacterial pathogen so far that replicates in a very large compartment, as I mentioned before. As you can see here, in green you have Coxiella, and in red you have LAMP1, which is a marker of late endosomes and lysosomes. And as you can see, Coxiella accumulates in this large environment. However, the fact that this was, until a couple of years ago, an obligate intracellular bacterium severely hampered research on Coxiella because we couldn't do any genetics on it. So as a result, the information that we have today on the bacterial effectors that Coxiella uses to manipulate the signaling pathways of the host remain largely unknown. And virtually no virulence factors of the bacterium have been identified so far, although some papers on Coxiella are still already emerging. So what we know about the cell cycle of Coxiella is that there's a difference between the way phase 1 organism and phase 2 organism enter the cell. Phase 1 organisms are associated with a big rearrangement of the active cytoskeleton, whereas internalization of phase 2 organism is more similar to a zipper mechanism that is not associated with large rearrangement of the cytoskeleton. Whichever the way bacteria enter the cell, they are fined into a tight-fitting early endosomal compartment that is positive for typical early endosomal markers such as RAB5 and EM1. It has been reported that early during infection there's a transient recruitment of autophagosomal marker, and it has been suggested that interaction with the autophagosomal pathway is a way for Coxiella to temporarily stall endosomal maturation, and this would allow Coxiella to prepare the endosomal compartment where it can grow. A couple of days post-infection, bacteria are found in a much larger endosomal compartment where replication still has to occur. So actually Coxiella grows the compartment before replicating within it. And the internal pH of this compartment now is dropped to approximately a pH of 5, which is fundamental to activate bacterial metabolism that now starts to secrete a number of bacterial effectors through a type 4 secretion system, and I will come to that shortly. And at this stage, the Coxiella-containing vacuole is positive for RAB7, LAMP1, flotillin, and all the other markers of late endosomes. 6 days post-infection, this endosomal compartment is much bigger in size, as you can see from this video that shows you epithelial cells infected with Coxiella GFP tag. And as you can see in phase contrast this large vacuole, and you can see that from day 3 to day 5 post-infection, the amount of bacteria replicating within this compartment increases, and you can follow this by fluorescent microscopy following a GFP signal. And importantly, at the end of the 6 days of bacterial replication, what has been reported is that Coxiella is not does not lead to a lytic infection, in the sense that bacteria does not escape the infected cell by lysing the infected cell. Actually, bacteria re-enter a kind of a stationary phase, a dormant phase called small cell variant, and actually they remain within the infected cell, and the infected cell can be cultured as a normal cell, which also poses the question of how actually Coxiella spread the infection from one cell to the other. And to persist within the infected cell, also Coxiella inhibits the apoptosis of the infected cell to protect the cell from dying, which is not favorable for the bacteria. So, as I mentioned, nothing is really known. I mean, very few things are known of how Coxiella can do all this, and the few information we have derive from the fact that Coxiella uses a type 4B secretion system to inject bacterial effectors in the cytoplasm of all cells. And it's been reported that the Coxiella type 4 secretion system is highly homologous to the type 4 secretion system of Legionella pneumophila, which is not an obligated cellular bacterium. Genetic is available for Legionella, and Legionella has been widely used as a model to study Coxiella infection. Actually, there's been a few cases where Coxiella effectors have been impressed by Legionella pneumophila to try to determine the role of Coxiella effectors. But this, although this was really a remarkable advance, it still limits our study to what we know about Legionella pneumophila, because the only bacterial effectors that have been identified so far, at least putative bacterial effectors that have been identified so far in Coxiella, derive from analogies of what we know in Legionella pneumophila. So it's not really a wide-scale identification of Coxiella burnetii factors. This similarity between Coxiella and Legionella has been used by a number of labs, and in one example, the lab of Jim Samuel in the United States have reported a list of 60 putative Coxiella effectors that have been tested in the Legionella pneumophila for their type 4 secretion system dependent secretion. And so that we have at least an idea of what could be the factors that Coxiella uses to replicate within the cell. But as I said, this is very limited type of information. Now, a couple of years ago, in the laboratory of Robert Heinzen in the United States, they have developed a very nice medium that now allows us to cultivate Coxiella outside the host. So it's not anymore an obligated cellular bacterium. This is an acidic medium that basically replicates the environment that Coxiella finds within lysosomes. This medium allows, of course, the exenic culture of Coxiella burnetii in microaerobic conditions. We can now culture Coxiella in liquid and solid broth, and more importantly, we can now genetically manipulate Coxiella and isolate bacterial mutants using this culture techniques. And these strategies have been, this technique actually has been used already by the laboratory of Robert Heinzen, but also from the laboratory of Craig Roy, that have started to mutagenize Coxiella and isolating a few mutants in genes that are essential for the type 4B secretion system. And they have given formal evidence that the type 4 secretion system of Coxiella is functional and necessary for bacterial replication. This is the example of the paper published by the Heinzen laboratory, but similarly, as I mentioned, the Roy's laboratory have published similar examples. Now they can cultivate wild-type Coxiella and the type 4 secretion system mutant in the exenic medium, and they replicate with similar kinetics. But when they infect the cells, they can see that the wild type grows within the infected cells, without the mutant that has a mutation in the type 4 secretion system fails to replicate. Now what I think, and here you can see also by microscopy, in blue you have the nuclei of the infected cells, you can see in red the vacuoles that Coxiella generates within the infected cells, and in the mutant of the type 4 secretion system, Coxiella fails to develop these vacuoles. Now what I found interesting is that they found a way to inducively complement the mutation, and what they saw is that if they infect cells with the type 4 secretion mutant, this gets delivered to a LAMP3 positive compartment, so it's possibly within a lysosome, but it's not dead, it's still alive, because if after 2 or 3 days of infection they trigger the complementation of the mutation, now the bacteria are able to grow again, indicating that even though the bacteria cannot replicate within the lysosome, it's still viable. And what I also find interesting is that they try to co-infect the type 4 secretion mutant with wild-type Coxiella, and the presence of wild-type Coxiella trans-complement the mutation of the mutant, which now is able to grow as if it was a wild-type. And this is important for our studies that I will come to in a few slides. So what we want to do, we want to take all these techniques that have been developed and put them together to develop a high-throughput screening to identify as much as possible all the bacterial effectors that are important for Coxiella replication. So we identified a few steps of the infection cycle that we think that are fundamental, such as adhesion to the host cell and internalization, of course, vacuole biogenesis and intracellular replication, the inhibition of apoptosis of the infected cell, and also we're pretty much interested in understanding how Coxiella spread the infection from one cell to the other. Now we took these essential steps of the Coxiella cycle, and we are developing actually 2 lines of research. One that we call Q-Screen, by which we are generating a bank of transposon mutants of Coxiella phase 2. And we are using the host cell basically as a toolbox to infect with all these mutants and identify all the bacterial effectors that are fundamental for these different steps of the infection. In parallel, we are also developing another approach that we call Q-Interfere, by which we will infect cells with wild-type Coxiella, always phase 2. This time we will infect cells that have been treated with banks of genome-wide siRNA molecules or microRNA molecules, this time to identify the host factors that are fundamental for bacterial replication. And we believe that cross-comparing the results that we obtain from these 2 approaches would give us a very comprehensive idea of the host-pathogen interaction that occurs during Coxiella infections. Now this project has been delayed a bit. Now it's kind of ready to go, but we haven't really obtained the results from it. So what I'm going to concentrate my talk on today is the Q-Screen approach on the Coxiella mutants. And to generate a bank of Coxiella mutants, we have obtained from the laboratory of Bob Heinzen a HIMON1-based transposon, which we have modified by inserting the EGFP gene under the control of a strong Coxiella promoter, under 11. And we are electroporating this transposon in Coxiella burnetii phase 2, and we can now generate Coxiella mutants that are also fluorescent, and we can use exenic culture techniques to isolate all these mutants, amplify them in liquid culture, and we have adapted a technique of single primary colony PCR to identify the site of insertion of the transposon in all the mutants that we have isolated. And of course, all the mutants are stored in specific 96-well racks and stored for further high-throughput screenings. Now, Coxiella has approximately 2,000 genes, which means that to have a comprehensive base, we're aiming, to have a comprehensive bank of mutants, we're aiming at obtaining approximately 6,000 mutants. And to date, we have approximately 3,000 clones that have been isolated, stored, and are being sequenced and tested. And what follows is the result that we obtained from the first approximately 900 transposon insertions that we have isolated, sequenced, and tested. Now, as you can see, this is the genome of Coxiella burnetii. The green dots represent all the transposon insertions that we have isolated and characterized so far. As you can see, luckily for us, the insertion of the transposon is pretty homogeneous throughout the genome, which is a big advantage for us. There's a few hotspots of transposon insertions where we have a lot of green dots, like up here, 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 and here. And actually, this one here is pretty interesting because in this area of the genome, we have all the genes that code for the type 4 secretion system. So we've been able already to isolate a large number of mutants in the type 4 secretion system. Now we have all these mutants and we have to find ways to rapidly screen them. And what we came up with is to use actually the fact that these mutants are fluorescent. So what we do is we use 96-well plates where we culture epithelial cells. And we are infecting these epithelial cells with our fluorescent mutants. And we're using a microplate reader that reads variations of fluorescence in the GFP signal as a marker of bacterial replication or non-replication during 6 days, 7 days of infection. Now this is pretty efficient, but this is the general kind of data that we obtained from one single plate after 7 days of infection. It's pretty complicated to interpret, but to validate our assay, we isolated a few mutants that we had in the beginning. And if this is the curve of the growth curve of the wild type in infected cells, if we compare this to 3 independent transposon insertions that we have isolated in the gene DALT-A, which is an essential gene in the type 4 secretion system, we can see that all the 3 mutants fail to replicate within the affected cells. And similarly, we also have isolated 3 independent transposon insertions in the cytochrome C oxidase gene, which is essential for bacterial metabolism, so nothing to do necessarily with host-pathogen interaction, but again, a nice control. And also these 3 mutants fail to replicate within the cell during the infection cycle. So this is a very nice way of following day by day the replication of every mutant on culture cells, but what the information that gives us is a quantitative information and not a qualitative information. So we know that some bacteria will grow, some other will not grow, but we don't know why this is happening. So at the end of the 7 days infection cycle, we're fixing our cells and we're switching to automated image analysis to try to understand why some mutants don't grow inside the cells. So the idea is to develop assays that can allow us to obtain as much information as possible from one single immunofluorescence image. So imagine that this is an image of an infected cell. From this infected cell, we have developed a few assays that will allow us to understand how many infected cells we have in our samples, what is the size of the Coxiella vacuole in each mutant, an approximation of how many bacteria we have in each infected cells, and by evaluating the morphology of the nucleus, we can also estimate how many cells enter the apoptosis in each kind of infection. And of course, if we want to do this on a large number of cells, we have to move to high content fluorescent microscopy, and we are lucky enough to have an automated microscope at the institute, at the imaging platform of the institute. So we can bring our plates at the imaging platform, we can just program the robot, and it does everything automatically. And we have programmed the microscope to have to image actually 6 independent fields in each well of a 96-well plate. And for each image, we're acquiring the transmitted height, the GFP signal, the DAPI signal, and we're also trying to develop ways of acquiring the end-cherry channel where we can have markers of the endosomal compartments of the cell. So this means that if we have 3 plates for triplicates in each screen, we have approximately 2,000 images per plate, and when we are lucky, we are obtaining 14,000 images per week to analyze. So of course we cannot do them one by one, and we're using an amazing software. It's an open source software that's called Cell Profiler, and what this software does, we can actually program it to isolate the different compartments in this image. So in this image, this is a typical image that we obtained from the microscope. We have the Coxiella vacuoles in green and the nuclei in red. We can tell the software to separate every channel, to segment all the compartments, and to extrapolate a number of features from each compartment. And the protocol we're using, we are extrapolating approximately 10 features per cell over an average of approximately 12,000 cells per mutant, which means that statistically it's pretty relevant. And this is done all automatically, but of course it means that we're going from having a lot of images to a lot of numbers. And we spent a lot of time in developing an automated Excel spreadsheet to perform all the required statistical analysis of all the data that we obtain. And what we can do with these data, an example of the things we can do, we can actually plot the average colony size per mutant in pixels versus the average number of Coxiella colonies per infected cells. And we end up having charts like this, and with a bit of statistical analysis, we have identified a region where we have mutants that do not differ too much from the wild-type Coxiella. This is another image that we get from the microscope. In red again, you have the nuclei, and in green you have the Coxiella vacuoles. And then we have a region of our chart where we have mutants that actually sometimes produce more vacuoles per cell, but of a size that is significantly smaller than the wild type. And this, for instance, is the case of the Daute mutant that I showed you before. This, as I mentioned before, is a mutant that is a mutant in a gene that is essential for type 4 secretion. And if you zoom into this area, you can see that actually bacteria enter the infected cell, but they could not develop the big, large, characteristic vacuole of Coxiella within the 7 days of infection. And the reason why we think we have more than one vacuole per cell is that in a normal situation, the Coxiella vacuole is highly fusogenic, whereas if you prevent type 4 secretion, then Coxiella will grow into isolated vacuoles. And once again, we have another area down here where we have simply very few Coxiella vacuoles per cell of a smaller size. Now this is a a very extreme case of a mutant that is down here, a mutant that we call 2143, just because we're giving numbers to every transposon that we have. And as you can see in this case, you just have the nuclei of the infect of the cells, and you only have a couple of vacuoles here. Now this is a phenotype that could be due either to a defect in bacterial invasion of the infected cells, or a defect in bacterial survival within the infected cell during the 7 days of infection. And in this specific case, we did a little bioinformatic analysis, and actually we found out that this transposon insertion falls into a gene that codes for a lipid of the membrane of Coxiella. So probably in this case, we're dealing more with a case of survival of the bacterium within the infected cell. By scanning all these mutants, we actually find mutations that have an impaired internalization within the infected cell. Now, and then last but not least, we also have isolated a few mutations that actually leads to bigger Coxiella vacuoles. So it seems that mutation in these genes actually is favorable for Coxiella replication. And curiously enough, this is another transposon insertion in a gene of the type 4 secretion system, ICMB. We honestly haven't gone much further with this, but we found it was interesting. And if we superimpose this with all the mutants that we have obtained from the type 4 secretion system mutants, we can see that they mostly fall into the areas where we have a defect in replication. That confirms what have been reported so far in literature for Coxiella, but also for Legionella that has a similar system. As I said, we have the ICMB mutant here that seems to grow even better, and an ICM mutant here that doesn't seem to play any major role in Coxiella replication. Now, what we also figured out pretty rapidly is that we now are accumulating a lot of information coming from the sequencing of all our mutants, the site of insertion of our transposon, the characterization of the genes that has been mutated, the phenotype of intracellular growth during the 7 days of infection, and also the morphological phenotype that we obtained from the microscope. And we had to find a way to integrate all these data together, so we have developed this nice database, which is based on another program, this time unfortunately not open source, that is called FileMaker. And what this database now allows us to do is just to search for a gene in the Coxiella genome, and we'll turn up if we have a mutation there, or on the other side we can actually look for a mutant and see which gene is mutated. It tells us where it is stored in our -80 freezer, how much we have, if we have tested it, if we have information on this gene in the literature. So it's a very powerful source of information that it's growing day by day as much as we get more information about our mutants. But what is also important is that we can now ask this database to only give us information on a certain kind of mutant, so we can actually ask the software to give us also only the information of mutants that have a significantly impaired replication within the cell. So we can actually focus our analysis on these mutants here that have a significantly decreased replication within the infected cell. And by doing this, we have identified approximately 116 mutants. We did a bit of bioinformatical analysis in all these mutants, and we found out that we have a number of these mutants that have transposon insertions in genes that we consider essential to bacterial metabolism. So we tend to discard those, not because they're not interesting, but just because we're more interested in identifying genes that code for proteins that are involved in host-pathogen interaction. So of course we believe that if we knock down the energy production of bacterium, this will simply not replicate within the cell. But we have also identified a number of interesting mutations. We have 19 mutation in genes of the type 4 secretion system that are here illustrated in red, and they all have a major impact in bacterial replication. We have, again, as I showed you in the beginning, the gene of the cytochrome C oxidase that when mutated has a severe impact on bacterial replication. And as a nice control, we also have a mutation in PMRA, which is a master regulator of the expression of all the genes of the type 4 secretion system. Now, we are retaining all these genes, and we're trying to develop assays to better investigate these mutants. And so we started with an analysis of the mutation that we have in the type 4 secretion system. This is the list of those that we have already mutated, and on the side you can see how many mutants we have isolated for each gene. So in some cases we only have 1 mutant, but in some other cases, such as the dotted mutant, we already have 8 independent transposon insertions. And if we analyze all these mutations that we have identified, as expected, a large number actually of these mutants that have severe impact in bacterial replication. But we also have identified a couple of mutants that have 50%, let's say, inhibition of bacterial replication, which is pretty interesting to us. These are mutations in .B and ICMS genes. And we also have identified a couple of mutations, as I showed you before, that doesn't really severely affect bacterial replication. So we were particularly interested in these two ones because we thought, well, either it's a problem in the way we evaluate our results, or it actually could be an important phenotype. So we went back to the information that we had by the microscopy analysis, and we can actually see that the wild type grows happily, the .A mutant fails to grow as expected, and the .B and ICMS mutant actually managed to grow to some extent. But if you zoom into the images, and well, the resolution is not amazing because these are the images we acquired from the automated microscope, but you can see that actually these mutants replicate to some extent, but they fail to generate this large vacuole that is characteristic of the wild type. So it could suggest that these two type 4 secretion system protein could be involved in the fusogenicity of the Coxiella vacuole. So this is something that we're trying to investigate further nowadays in the lab. Now, concerning all the other mutants and genes that we have identified, we have a few. There are mutations in genes that code for eukaryotic-like domains containing protein, hypothetical proteins, and other intergenic proteins. And in order to identify those that are really relevant for us, we are using this approach of co-infecting the mutants with the wild-type Coxiella. Now we have wild-type Coxiella, which is expressing the red fluorescent protein and cherry, and we are co-transfecting cells with our transposon mutant that is GFP. And we are exploiting what has been observed in the laboratory of Paul Heinzen, and we assume that if the co-infection doesn't transcomplement the growth of the mutant, it should suggest that we have mutated a metabolic gene. Whereas if the co-infection of the two mutants transcomplement the growth of the transposon mutant, then we'll probably have mutated an effector genes, and the presence of the wild type could complement the lack of this gene in the mutant. And we have performed a couple of control experiments, and as you can see, these are cells infected with the .A mutant. But if we now co-infect cells with the wild type and cherry and the .A GFP, we can see that the .A GFP now manages to grow similar to the wild type. So we're using our image analysis software to segment these Coxiella vacuoles, and the software can give us information of how frequent this transcomplementation is, what is the size of the vacuole, the wild type and the mutant vacuole, and have a lot of information on the nature of these genes that we are mutating. Now, another important thing that we need to know when we study the role of an effector is, well, first of all, whether this effector is actually secreted by the type 4 secretion system, and where does it go within the infected cell. And there's a lot of assays that are already available, such as the beta-lactamase assay, but we are trying to implement these techniques by developing, adapting actually an existing technique to the study of Coxiella effectors. This is the split GFP system that has already been used in the Salmonella studies. And briefly, what a laboratory in the US have done, they have managed to split the GFP molecules into two. So the GFP molecules is composed of 11 beta barrels, and what they've done, they have actually split it in two. One contains the barrels from 1 to 10, and the other one is just 11th beta barrel, which is pretty small and is compatible with type 4 secretion. So we can now tag proteins with the 11th beta barrel of GFP, and when the 11th beta barrel recognizes the other barrels of GFP, now they reconstitute the full protein, and we have GFP fluorescence. So what we are doing, we have produced a stable epithelial cell lines that expresses cytoplasmic GFP 1 to 10, and we are actually these days tagging a number of Coxiella effectors with the 11th beta barrel of GFP, and we are expressing these constructs into Coxiella and cherry. So we will infect cells, and we will have Coxiella and cherry generating red fluorescent vacuoles. And if the protein we are following is actually secreted in the host cell, you will recognize GFP, the barrels 1 to 10, and give a fluorescent light in the compartment where this effector sits. So if the bacterium secretes this protein in cytoplasm, we will have the whole cytoplasm of the cell that turn up green. If this effector is secreted and is localized, let's say at the bacterial vacuole, then we will have the vacuole that will turn green. And so we're doing a few controls, but this technique is really promising, and it allows us to follow by light imaging, actually in real time, bacterial secretion, and to identify at the same time the sites of localization of bacterial vectors. So this is all an ongoing kind of screen that we have in the lab. Now another thing that we asked our software is now to give us all the mutants that have an impaired internalization within the cell. And this time we have identified 69 mutations. Once again, we have a number of genes that are probably involved in essential bacterial metabolism, so we're keeping them there but not really going after them. But what's important in this case is that we have identified 3 independent transposon insertion in mutant 208, 175, and 27 in the same gene that actually codes for a protein that is potentially involved in receptor-ligand interaction. So this was, of course, very interesting to us, and we are trying to characterize the gene further. So we have analyzed the sequence of this gene, and it contains a transmembrane domain, and the predicted structure of this domain is like this. So we have a transmembrane domain, a pretty small actually extracellular domain, and a very large intracellular domain. And so we went back to, we call this gene temporarily Coxiella internalization protein 1. We will see if actually it is an internalization protein, but we hope so. And we analyzed the gene that has been mutated just to be sure that it was not present into a series of genes, but actually is an isolated gene. So actually the effect that we're seeing is really dependent on this gene. And if we zoom in into the sequence, these are the site of insertions of our transposon. We actually recently identified a 4th transposon insertion in this gene, but we still have to see what effect it has. So we went back to the data that we have obtained from our microfluidic trigger assay, and we could see that all the 3 independent transposon insertions that we have isolated from this gene severely affect bacterial replication. And so we are focusing now, for simplicity, on one of these mutants, the mutant number 208. And by PCR, we have confirmed the insertion of the transposon into the gene that normally is 747 base pairs. But if you PCR the same gene that contains the transposon, we have a shift in size that is compatible with the insertion of the transposon. And by PCRing with another primer that is inside the transposon, we can actually tell the directionality of the insertion of the transposon. And we're now currently doing the Southern plot to further this insertion. And so what we wanted to control then is whether the failure of replicating within the cell actually concerns bacterial internalization. So we did a few tests comparing the wild type, and what you can see here are infected cells stained with actin in red. And you can see bacteria that are double labeled because we are using a fluorescently tagged wild type, so it turns out in green, and we are labeling extracellular Coxiella before permeabilizing the cells. So the anti-Coxiella antibody will only recognize bacteria that are sitting outside the cell. So those that are outside the cell will turn out blue-green, and those that are inside the cell will actually be only green. And if we compare the wild type to our CIP1 mutant 208, we can actually see that in this case, most of the bacteria are actually sitting outside the cell. We have one bacteria here that made this way in. And if we compare this to another mutant that has a transposon insertion into an intergenic region and doesn't have any significant morphological phenotype, we can actually demonstrate that this effect is not simply due to the fact that these bacteria have a transposon inside, because this is exactly the same transposon but into another region of the genome and doesn't have any effect on bacterial internalization. So we are going back to our image analysis software, and this time we're asking the software to identify all the bacteria that are sitting outside the cell, and the software is putting a nice red circle around them. And now we ask the software to identify all the bacteria that are inside the cells, and we have a white circle around those. And if we quantify this over approximately 8,000 bacteria, 8,000 bacteria analyzed in each sample, we can see that wild-type infections, and in the case of the controlled transposon insertion, bacteria gradually invade the cell, whereas if we're looking at the mutant in CIP1, these mutants fail to enter the cell. So we really believe that this protein is involved in bacterial internalization, and we're now in the process of purifying this protein to coat beads and do all the additional controls that will actually formally prove that this could be actually one of the ligands that are important for Coxiella internalization. Now, last but not least, we're also, as I mentioned in the beginning, analyzing the protection from apoptosis phenotype. And there's a couple of information that we can also obtain from the first screen that I showed you before. And simply, it's a pretty indirect method, but it kind of works. We're actually quantifying how many infected cells survive 7 days of infection, assuming that if we have a mutation in a gene that is involved in protection from apoptosis, we should have less infected cells at the end of the time course. And we have identified a number of mutants that actually lead to a severe decrease in the number of surviving infected cells. But since, as I mentioned, this is a pretty indirect method, we are doing the final controls for another screening that is almost ready to go, by which we are infecting our cells with our Coxiella transposing mutants. And after a couple of days of infection, we're treating cells with staurosporine that induces apoptosis. And the result of staurosporine is that the nucleus is fragmented. So we assume that if the cells are infected with Coxiella that is able to protect the cells from apoptosis, we will have normal nuclei. But if we now infect with Coxiella that have lost the capacity of protecting the cells from apoptosis, then we will have fragmented nuclei. So we tested this in a few controls, and now we can tell our software to identify the normal nuclei versus the fragmented nuclei. And if we have non-infected cells not treated with staurosporine, most of the nuclei are of course normal. If we now treat non-infected cells with staurosporine, most of the nuclei are fragmented, indicating that cells are apoptotic. If we now infect cells with wild-type Coxiella and treat with staurosporine, the percentage of cells in apoptosis is significantly decreased. But if we now infect cells with the Delta mutant that is failed in secreting effectors by the type 4 secretion system, the degree of protection from apoptosis is significantly increased. And these are the quantifications. So we believe that this screen is kind of ready to go, and we can actually rescreen all the mutants that we have obtained, trying to identify mutants that are impaired in protection of apoptosis. So to conclude, the exenic growth of Coxiella has opened the way to large-scale genetic manipulation. We are generating the first clinical GFP tech Coxiella transposing mutants. We are setting up high throughput assays for the unbiased identification of Coxiella virulence factors. And we have so far identified a transmembrane Coxiella protein that is possibly implicated in bacterial internalization in epithelial cells. We have a number of other ongoing projects and future directions to take. Of course, we will continue and complete the generation and the screening of the Coxiella bank of mutants. We want to now validate all the candidate genes that we have identified so far. We have an ongoing collaboration that aims at further characterizing the transposon insertion in intergenic regions of the genome and identify those insertions that have a phenotype in bacterial growth in order to see if we can identify non-coding RNAs in Coxiella genome. And we're also in collaboration with other laboratories trying to develop non-mammalian animal models to study Coxiella infections. And we have a nice collaboration in the UK to use Galleria melonella larvae to study Coxiella infections. And in Nîmes, close to Montpellier, we're trying to use zebrafish to study these infections, as in order to have a readily available and semi-high throughput system to screen all the mutants for kind of in vivo infection. And as I mentioned, we have more screens ready to go, the protection for apoptosis phenotype, the siRNA microRNA screens that I will talk to you about before, and the small molecule screen that is being carried out in collaboration with Sanofi. I think this is, I really won't have time to go through this, but basically we're trying to infect cells with wild-type Coxiella and treat all the cells with a bank of molecules owned by Sanofi in order to identify those molecules that prevent Coxiella replication without being detrimental for the infected cell. And with this, of course, all that I've been showing you now is not specific for Coxiella, but can be applied in the future to other bacterial pathogens. And we have a few in mind, such as Brucella, Coxiella, Salmonella, type 3 mycobacteria. And they all share a number of common features with Coxiella. And in the Host Institute, there's a large, high-capacity BSL-3 platform that is just being built, and we believe that once we have understood something about the Coxiella infection, we can actually try to apply all this system to the study of other class 3 bacterial pathogens. Now, I would like to thank the two heroes that chose to help me out in this task, because we're just 3 so far in the lab. We recently had the addition of a couple of master students. And of course, I would like to thank all the supporting agencies, the Aveniarty program that allowed the start of all these projects, Sanofi that is contributing to our research, We obtained a Marie Curie grant for the project of Eric, postdoc in that, and we also received funding from the Infectiopol Sud to acquire equipment for our studies. And we have a lot of important collaborations to actually allow us to study so many different aspects of the Coxiella infection, just being free in the lab. And with this, I think I will stop and move.
Thanks a lot. Question? Je peux poser en français.
C'est beaucoup plus simple. Deux ou trois questions. Premièrement, parce que je ne connais pas du tout ça, comment s'assure-t-on qu'il n'y a qu'un transposon par Coxiella et qu'en réalité, il n'y en ait pas 3 ou 4 ou 5?
So, can I answer in English?
Yes. I can answer in English.
It's okay. So we asked this question in the beginning. So what we did is actually what in the lab of Bob Hines and they did in the US and is actually working out pretty good, is that the transposon and the transposase are on 2 different plasmids. And the transposase is on a suicide plasmid that gets lost during bacterial replication. And the rate of Coxiella replication is so low that actually we're 99% sure that we only have 1 transposon insertion and that transposon will stay where it is. Now, we happen to isolate a few mutants that have maybe 2 or more transposon insertions. Now, we do not know if that was due to multiple transposon insertions or to the fact that we were not isolating actually a clone. Because what I didn't say is that these bacteria grow on agar, but they do very, very small colonies. So sometimes, but when we sequence these mutants, actually it turns out pretty clear if it's a single insertion or a double insertion. So we just discard these suspect double insertions or triple insertions, and we just follow all the others because we isolate so many that it's not a big problem.
This strain of Coxiella has plasmid, so
yes,
so the transposition is always in the chromosome.
No, we actually have a few interesting insertions in the plasmid as well. I didn't actually, I should have put also an image of the plasmid. Not as many, well, not as many as in the chromosome, but that also is, of course, due to the fact that the size of the chromosome in the plasmid is significantly different.
Last question. What about the transposition in intergenic regions? Because you saw the effect. So what does this mean?
So it could mean either the annotation was not accurate, but we don't think it's that thing, or it could mean that we're actually hitting a region of the genome that actually codes maybe for a non-coding RNA or small RNA that could have a role in infection. And this is actually, we're actually going after this because nowadays there's a lot of techniques that allows us to identify all the non-coding RNAs or the small RNA in a bacterium, but actually to identify those that have a role in infection is not so simple. So we're actually joining forces with other laboratories that are identifying non-coding RNAs. If we can contribute by saying, well, we have a mutant transposon, a mutation in this region that has a role, that could actually lead to the identification of relevant ones.
Est-ce qu'on peut considérer que les gènes qui ne sont pas touchés par le transposon, qui ne sont pas touchés par les transposons, qui ne sont pas sélectionnés sur le milieu axénique, sont des gènes essentiels?
Could be. It could be. Either, well, I think even if we isolate 20,000 mutants, there will still be some genes that will never be able to I don't know if actually we'll never be able to mutate. Maybe we are mutating them, but as you said, the mutant is mutated as an essential gene, so we'll just not grow with an exotic medium. But in a way, for us, for the questions we're asking, it's kind of an advantage, because if it's an essential gene, it's probably not implicated in those pathogen interactions, so we're just selecting it out automatically. Actually, I expected that to be more efficient, because actually we're isolating a lot of these mutant genes that I thought they were essential, but actually it turned out that they're not really essential. Actually, it's interesting, because for instance, in the case of the cytochrome C oxidase, it grows very well in exenic medium, but if you put it in the cell, it doesn't grow. So there's something also to study these kind of things. So far we're trying to focus a few things, but...
Do you have any mutants which is growing faster than your wild type control?
In examic medium is actually kind of difficult. So we have a very nice way of measuring the concentration of each bacterial mutant in each tube that we amplify to be sure that we're always using approximately the same MOI for infections. Inside the cells, we do have those mutants that generate larger vacuoles. Now, is that significant? Is that just by chance? But there are not that many, but there are some. So in this case, we have an ICMB mutant. We don't know what ICMB does in the context of the type 4 secretion system. If it's a negative regulator, we can imagine that that's a pretty naive hypothesis, is that if you take out an inhibitor, then you have a secretion of more effectors, the dipole gets bigger, but that's just one of the many hypotheses. So, but yes, we do have a few that actually grow better.
Tu sais ce que je vais te demander déjà, mais tu as fait de la phase 2, là. Comment tu vas faire pour le transposer sur la phase 1?
So we have, now that Bob Heinzen has published this technique of mutating the phase 1, we believe that that could be used to generate mutants in phase 1, or other techniques that could be available. Because to my knowledge, at least, if we then amplify phase 1, it will not immediately convert to phase 2. So there's still a bit of
no, but it's down virulence.
A window of opportunity. Then to test virulence, well, the idea is actually to, well, either reuse bacterial infections in vitro, or then use animal models. But the idea is that it's so simpler, it's so much easier to work on phase 2 now. And then we just want to turn back to phase 1 only when we have a more precise indication of a few factors that we want to go after.
And if you compare the sequence from the secretion system phase 2 and phase 1, it's similar, it's exactly the same.
The mutants? We have
just the wild type, already the wild type. If you take the genome from the wild type, phase 1 and phase 2, and you compare the secretion system sequence,
I think
into these forms.
Oh, I think it should be.
No, this question because you say that dot B and ECMS decrease the size of the vacuole, the mutants, and the replication.
They're less effective in replication, but they grow within what it seems to be smaller values.
Yeah, and it looks like what we have in terms of phenotype with the phase 1, for example. But can be an alteration of the genome which defines the virulence of the phase 1 versus the phase 2, and this effect is this difference in the secretion system.
It could be, but again, phase 1 doesn't do multiple small. It makes one small. And what we see are multiple small. So I think it's more dealing with the fusion between the two, well, the many vacuoles.
Yeah, very.
So I don't know if it's exactly the same thing.
And well, you didn't try to check your mutant, compare your sequence. No, you cannot. No, I'm still thinking about the phase 1 because since this secretion system, there is not only secretion system, but one of the factors which is important to with virulence for bacteria is the secretion system. Since this secretion system is present in both phases, I still don't understand how it can define part of the virulence. So there is some difference in the secretion system, and this is what I would like to know. What is the difference?
Well, the study we wanted to do so far is just to try to give a more specific role to the different components of the type 4 secretion system on phase 2. Then once we have understood something more, because now it's really nothing is known phase 1.
No.
Then we can try to transpose this to phase 1 and see what is the same, what is different. But I think so far it's important to understand possibly every component of the type 4 secretion system, what's its role, what it does, and so on and so forth.
But it will be interesting to match the genome from phase 1 and phase 2, different strain. Anyway, it's not
what I have
in the dot system, the ECM system.
Well, what I have stressed, but of course, our aim is actually to give a role to define gene in the phase 2, and then the last experiment is just go back to phase 1 to see if we can replicate the same.
Merci.